Okay, so this will be discussed not in detail, of course, because this is not something a biotech class to discuss the detail of antibody, DNA, or aptamer, but at least we understand how it works, how antibody works in the development of biosensor, how DNA works, how aptamer. So what is aptamer? Okay, so those are very important that we are going to discuss today. Yeah, the introduction only and the principle. Okay, so if you don't understand this first, it's quite difficult to follow the concept of sensors. Okay, so let me finish this one slide only before we take a break. This one slide, okay? Slideshow from current. Okay, right. Okay, so the first thing, as I asked you just now, if you have a smartphone and then you want to measure your oxygen level. Yeah. So it's very important to measure oxygen level. Okay, let's say, meaning that the first thing, so this is the whole component of sensors. Okay, particularly for chemical and biosensors. So now, as for the examples, oxygen to be detected. So oxygen will be what we call as the analyte, the one that we wish to measure. Okay, so analyte can be anything. You wish to measure glucose, you wish to measure virus, bacteria, protein, or drugs. So those are what we call as analytes. So easy to say, analyte is what you want to measure. Okay. Now, of course, whatever that you want to measure must be in certain units. Okay. So for example, if it is oxygen, it is in percent. And you understand it in percent, 90%. If it is below 94%, you have to worry about it. If it is 98%, then it's fine. If it's 100%, it's good. So the concentration unit of the analyte is very important for you to think about before proceeding in development of sensors. Okay, so the next one is, okay, this is the analytes. Okay, let's say it's oxygen. And this is what we call as receptors. Okay, it is written there is bioreceptors. Depends if it is biosensors, then the receptor is biochemical. But if it is chemical sensors, then the receptors are chemical reagent. Yeah, that depends. But one important things about the receptors, okay, it needs to have, if you look at what I circled, okay, let's say the sample analyte, okay, where is, let's say this is the oxygen, right? I hope that you can see the shape that it represents oxygen is the diamond shape, okay? So this is the oxygen. The rest, like octagon or hexagon, oval shape, rectangle, they are not oxygen. They are, yeah, a matter. A lot of, because if you're measuring blood, oxygen in blood, of course there are other matters in blood as well. They are CO2, they are blood cells, they are plasma, they are a lot of things, a lot of other matters in blood components, of course. So you can imagine that we want only, okay, we want only oxygen. You can see that oxygen interact with the receptors. This is the receptors. Okay, so be it a chemical reagent as the receptors, or biochemical reagent as the receptors, it has to specifically interact with oxygen only. Okay, so this is something that's very difficult to achieve. Very difficult to achieve. And beyond this interaction, okay, I have to draw it here, the diamond shape, okay, and then you have these receptors, okay. So you can see that other shapes like oval, rectangles, hexagon does not react with the receptor. So you can see that this diamond react with the receptor, and this reaction, okay, will have some effect, okay, will have some effect on the transducer. Okay, transducer is something that we are going to discuss after your break. So now what you should understand is the meaning of analytes is something that you wish to measure, because biosensors sensors is about measuring, it's about detecting. Like we walk to the shopping complex and the door open because it detects somebody so that the door open. So sensing is about detecting. So that's why we need something that we want to detect, which is the analytes. Okay. The second one that you need to understand is how it detects. So based on the automatic door just now, is based on resistance, because you are some solids, and then you block the sensors. So there is a resistance. So the sensor tells the door to open. So that's how it works. So for biosensors, among the matters in the blood, and you wish to detect oxygen, which is represented here as the diamond shape. So you need to have this chemical reagent or biochemical reagent that only respond to oxygen. Because if the receptor responds to like CO2, urea, or other components in the blood, then it's not going to be a good sensor, because the reading will be not accurate, because it does not represent the existence of oxygen. Okay, so I hope that by now you understand until this part. Okay, so of course the bio and the chemical reagents, there's a lot of bio and chemical reagents that exist and that has developed for sensors. But for these lectures, we only discuss the three, not these lectures, this subject, we only discuss DNA, antibody, and aptamer. There's a lot of other things being developed, but we don't have that capability to discuss all in this subject. Okay, questions before we stop for a break? Okay, so this sensors is very important because it sort of replace all the bulky machines. Okay, not to say replace, at least well, before this, when you need to detect drugs, you have to use chromatography, whether it's HPLC, or you have to use like GCMS. Imagine that you have to do that every time. Okay, so when we have an alternative, not to say to replace all those good machines, but at least an alternative. Okay, let's say you need to detect drug of abuse. Okay, you need to detect drugs anywhere during police operations. So you need to have something portable, okay, to detect there and then. Okay, and imagine the issues of contamination of river water. So if you have to bring back every time the samples from the river to the lab, and then make sure that just trying to get things, okay, what is inside the river, why is it affecting the water supply, and you want to trace back the samples, the contaminants to which industry. So this will be time consuming. If we have something portable, okay, handheld, you can detect there and then. So of course, our life is much easier. Okay, but developing a handheld sensor, a portable device, is not easy. As I mentioned, it requires three different fields of science: engineering, chemists or biochemists, as well as a software, which is IT now, IT assisted sensor. Okay, everybody. I think we take a break. So I'll see you around 8-ish. 8-ish mean 8:10, 8 10 after 8, or probably you can be here at 8. Okay. Okay. So you can bring your drinks, your food together. Okay.
Okay,
So
bro. Thank you so much.
Okay, Prof.
resume the as I mentioned just now, this subject does not teach you like the theory of IR, for example. Does not teach you the theory of GCMS, spectroscopy. Okay, because you have learned that in atomic spectroscopy. Probably some of you are taking molecular spectroscopy. I'm not sure. Or you have taken molecular and currently taking atomic spectroscopy. So you learn about the theory of IR, you learn about the theory of atomic spectroscopy, you learn about fluorescence spectroscopy, all about theory. So for this subject, it's about application. Means that whatever techniques that you have learned in the spectroscopy, for example, this is where you apply. Part of it is you apply, because for sensor means that you sense, you are sensing, you are detecting something, some compound. Okay. So of course, in order to understand the subject, you have to remember all those techniques. The one that you have gone through before, which is spectroscopy, or currently taking. Probably you are you are currently learning about fluorescence, or you are currently learning about IR or MS. Okay, so those techniques are going to be used. Some of it are going to be used in this subject. So you have to revise back, because I'm not going to discuss back the theory of fluorescence. I'm not going to discuss back the theory of UVVs, AS, ICP, because those are, I assume that you have gone through the topic. Okay, so now we have discussed just now up to this. Okay, and of course we are going to discuss detail of not during this lecture, on the receptors. Okay, so the receptor part for this class today is only for you to understand how it functions. It it interacts specifically with the analyte. So there is a function of receptors, and receptors can be in a form of chemical reagent. Okay, also it can be in a form of biochemical reagent. Okay, so it's different when we are talking about physical sensors. Yeah, physical sensors, I have given you the example of automatic door. Okay, so there is a resistance, so the door will open. Another example of physical sensors is, for example, smoke detector. Okay, there is smoke, there is heat detected by the smoke detector. So at certain level, you will hear the fire alarm. So those are physical sensors, okay, that I can think of. There's a lot of physical sensors. it is a sensor, but based on physical parameters. Okay. Now, the third part is transducer. So what is transducer? Okay, this is very important. All these components are very important in development of sensors. Remember that I mentioned about you need those from engineering. Okay, engineering to design all the circuits, all the hardware. Okay, and you need someone from bio or chemistry who understand the reaction. Okay, the specific reaction, the mechanism, and of course, we need software. Because if we don't have, if you see here, okay, as users, okay, we are public users, we only want to know the result. For example, if it is oxygen, we just want to know whether it's 98% or 96% or 90%, for example. We just want the final result. We do not want to do any calculation. Okay, if it is, for example, glucose with certain concentration, usually if it is above 7, then you must be worried about your glucose level, for example. So those are the final results, okay, that the users, the customers, or whoever is doing the analysis wish to know. Okay, you do not want to know about the calibration. You do not want to know about the calculation, whatever process that is inside the sensor. Okay, so that's why you need somebody to do the programming for you. Okay, if we are to develop a sensor, somebody has to do the calibration, built-in calibration, calculation automatically done. Only the results will be on the display, the final results. Okay, so when we talk about transducer, is the technique, okay, the technique in detecting the interaction. So just now I give you, for example, okay, let me move to your smartwatch, because those are quite common to not to say almost everybody, because it is expensive, but most people. Okay, so now you are measuring oxygen in blood, which is as the analyte. So oxygen is the analyte, right? Now, this is your skin. This is your skin, yeah? And this is the component, the blood component. So now you have oxygen. So I put the circle one as the oxygen. And there are others as well, probably CO2, probably urea, or other components in blood. Okay. Now, it's not urea. Now, how does the watch, yeah, detect the level of oxygen? Okay, so now you have this green one. So this is the electromagnetic radiation. I don't know whether they are using what kind of radiation. That is their trade secret. That is their secret. So when you wear the smartwatch, okay, and it comes to close encounter or close proximity with your skin, I don't know how close it can detect, or it has to be very close or touch your skin. I'm not so sure about their technology, but there is a radiation of certain electromagnetic wave that goes through your skin. And they, of course, have done some calculation before. Okay, this is, I can consider as physical sensor. This is not chemical or biochemical sensor. There is no reaction whatsoever, but the sensing is based on, you can see that the radiation, the green light here, okay, you can see the green light here is the radiation of the electromagnetic. Okay, so some pass through like this one, and some actually interact with the oxygen molecule in the blood. Okay, so they have done some software, some intelligent thing, so they can calculate this these interactions between the radiation, the electromagnetic radiation and the oxygen, and probably through the mathematical equation, they can come up with the percentage of oxygen in the blood. Okay, so this is actually a bit difficult because you can see that the electromagnetic radiation can also interact with other matters in blood. Okay, so there's a lot of I believe there's a lot of experiment, there's a lot of calculation, there's a lot of validation to make sure that the calculation is correct and giving you a correct, accurate percentage of oxygen. So what you can relate with the sensor component just now, I have highlighted, so the analyte is oxygen. And let's go back. There is no receptors, okay? There is no receptors because it's just physical. But there is interaction between transducer and the analytes here. So I believe the transducer is the electromagnetic wave. I don't know whether it's laser or what kind of electric wave, what is the lambda wavelength that interact with the oxygen. Okay, so here is the transducing effect. Okay, I'm saying transducer. Transducer is the technique. Okay, what happened is the transducing effect, meaning that the interaction between the oxygen molecule in blood with the electromagnetic wave. So there is some changes and it is calculated mathematically, and the results is in terms of percentage of blood oxygen. Okay, so you can see that you have analyte and you have transducer, which is affected by the oxygen in the blood, and then you have the final output. Okay, so go back to this is the basis. So now you have the analyte, which is the oxygen, and then you have the transducing effect between the electromagnetic wave, which interact with the oxygen molecule, and the changes, whatever transducing effect between the transducer and the oxygen molecule is showed in the signal result. Okay, so in this case, it's percentage. Okay, so now I'm gonna focus on explaining to you the different types of transducer. Okay, so now you have learned about spectroscopic technique, right? Okay. So now you have learned about, I believe, atomic spectroscopy. I teach some part of it, 5406. I don't remember. So what techniques that you have learned, regardless of whether you have taken spectroscopic technique, molecular or atomic, what techniques have you taken? Have you learned before? Anyone just give some ideas?
IR,
Yes, IR.
NMR,
Some more?
fluorescence,
Yes, fluorescent. I put FL. Okay. Some more?
AES.
Okay. AES,
Can we
AS,
say UV, Prof?
sorry?
UV?
Yes, UV. Okay. All right. Very good. Okay. So this is some of the transducers that you can use because we are talking about technique. Okay. Transducers are technique to detect, let me go back to the picture just now, to detect the changes. Okay. Because what happened just now, okay, based on your smartwatch, when the electromagnetic radiation, the green one, interact with the oxygen molecule in blood, there is a change in the intensity probably, or based on what kind of changes that you wish to measure. So the changes must be detected by a transducer. Okay, so this is the transducer that you can use, transducer technique that you can use, whether it's fluorescence, maybe UV, maybe IR, NMR, Raman, or this kind of transducers. But of course, some are difficult because, for example, if you choose to have NMR, we know that NMR is very big. There are some scientists who wish to miniaturize NMR. Okay, but yeah, it's difficult. And of course, we can use IR to detect the changes that happen when the analytes and the receptor interact. There is a change. So we can choose to have these techniques, these transducers, to detect the changes. But we have to be sure which will give us the best results. Okay. There are other techniques, for example, electrochemical technique, because this will result in current changes. Meaning that when there is an interaction between the analytes with the receptors, there is a current changes. So there is electrochemical technique that you can use to detect the changes. And these are all what we call as transducers, because it's transduce the changes into some readable signal, whether it's current, whether it's based on lights, whether it's based on absorbance or fluorescence. So the changes or the interaction will be translated into a signal. As I mentioned just now, fluorescence, absorbance, current change, or probably IR spectrums. Okay, so some are difficult. For example, NMR, IR, because IR use the light source. Okay, is expensive. Okay, NMR, the principle is complicated. So, so some techniques are a bit difficult to be used in development of sensors. So among the transducer that easily being incorporated in sensors are electrochemical technique, because it is based on current changes. Optical, which is probably absorbance, you see UV just now, fluorescence, or it can just be color change, just like the test strip that you have used for COVID test. It is based on color change, but this falls under optical technique. So this is transducer all about. It helps to translate the interaction into signal. Okay, because we have to measure the signal. We have to measure the interaction. Okay, we have to measure the interaction between the analytes and the receptors. Okay. Questions? I hope that you understand now transducers, yeah? Okay. It is written here, measuring the production change that occur with the reaction at the receptor, regardless if it is chemical or bioreceptor. Then transforming the interaction into a signal, useful signal, measurable signal. Okay. If it is color, then we can look at the colors. We can measure the colors. So if it is current, we can look at the current. If it is intensity of fluorescence, we can look at the intensity of the fluorescent changes as well as absorbance. Okay, so choosing the right transducer for the right chemical reaction or biochemical reaction is very important. Okay, the last part, as I mentioned, it has to be translated again to a readable signal into the concentration that is, of course, required by the user, because we are the users. We do not want to worry about the calculations. We just want to know the results. So that is what sensors are for. You want to know your glucose level. You want to know your oxygen level. You just want to know the final result. You don't want to know. You don't want to calculate anymore. Okay, that's the purpose of you buying, for example, glucose detector. You are buying a smartwatch to monitor your oxygen level because you want to know the final results. Okay, so these are all in general. Okay, all in general components of sensors, whether it's physical, chemical sensor, or biosensor. So remember, physical, usually you don't have these receptors. It's just, I'm not sure because I'm not really in physical sensors, but usually they don't have receptors. It's just that an interaction between the transducer and the analytes. Okay, they don't have the receptor part. Okay. So I hope the the part, the general part of sensor is clear to you. Now, when you look at your smartwatch, you look at the smartwatch differently. You have smartwatch, then you look at the smartwatch differently because now you know there are many parts that is working in that smartwatch. And then if you have glucose sensor at home, you're probably looking at the glucose sensor differently because now you know there is bioreceptors inside that glucose sensor, there is transducer, there is signal display, okay? And there are chemical reaction or biochemical reaction that happens when you drop your blood on the glucose tester. So probably you can look at the tester differently. Okay. There are other sensors. For example, if you have a test kit for COVID or for what other common just now, influenza. Yeah. You have your test kit now. You can look at your test kit for influenza or COVID differently because on the strips there is bioreceptors. And the transducer is optical. There is some nanomaterial that we put on the strips to give the colors. So that is optical based transducer. And then because it is yes or no, it's qualitative. Yeah? You just want to know yes or no, you have it or you don't have it. Okay, so the signal is quite simple. If there is no test line, you only have a control line, one line. So it's supposed to be no. If you have two lines, colored lines, it's supposed to be yes. So this is the signal readout. The display is quite simple. That's why it is cheap. Yeah? But cheap does not mean it always working accurately. Okay, so that's why we have different price. When you test for PCR, is different. The cost is different. If you only test for test strip, then the cost is different. So it all depends on the quality and the accuracy of the sensors. Okay. So now, if you have questions, you just can stop me. So I'm going to the last part of today's lecture, which is the receptors. Okay. So I'm going to focus some basic information on the bioreceptors. Okay. Okay. So as I mentioned, we have chemical, okay, and biochemical. So chemical reagent, there are a lot. Okay. If you remember your chemistry during your, I don't know, first degree, or probably, yeah, first degree probably. There are some chemicals that you use for spot tests. What does it mean, spot test? Let's say you want to detect mercury, or you want to detect chromium, okay, metals, okay. There are certain chemicals that you prepare, and when you react with chromium, you get green color, for example. I don't remember the spot test. You can Google spot test for metal. So what does it mean is the chemical reagents with all the specific names, okay, the chemical reagents, for example, EDTA, I don't remember, there's so many chemical reagents, okay. When they react with the certain metals, they change the colors, okay. So one of the easiest example is pH. Yeah, when you do a neutralization experiment, yeah, when you do acid-base titration, you want to know when to stop, meaning that you have achieved the neutralization or the end point. You have to put some chemicals, right, to make sure, okay, when to stop. There is color change. So when the color starts to change, probably from colored to non-color, or colored non-color to colored, you stop the titration. So that is what we call a spot test, and that is an example of chemical reagents receptors. They react with the, not to say very specific, they react to the products resulted from the acid-base titration, and they produce probably colored or non-colored. So depends on the reaction. So you have a lot of pH indicator, right? So those are examples of chemicals. And there's also a lot of spot tests for metals. Okay, you can react the unknown metals with the chemical reagents, and you got certain colors. For example, just now I mentioned chromium 6. You react with certain chemicals, and then you get green color. So now you know, based on the spot test, green color means chromium. So those are chemical reagents that works as receptors. It interacts sort of specifically to chromium, producing green colors. Okay, so probably there are other reagents that react with copper, producing certain colors. So those are chemical reagents. Unfortunately, chemical reagents, they are not that specific. So that is why it's difficult. You can imagine the chemical structure. Let's say EDTA is quite common. It reacts with many metals. So it's difficult if you want to just measure one metal, and when you add EDTA, EDTA is quite common ligand for detecting metals, okay? And you add EDTA, it will react with a different metal. So how are you going to detect a single metal, a specific metal inside the solution, because when you have samples, probably, for example, river water, you're not going to have a single metal only. You're going to have a multiple analytes in that sample. And if you choose certain chemicals, unless the chemical has been proven to be very specific, reacting with, for example, metal A that you want to detect, then you can use that as a receptor. But if that chemicals react with metal A, B, C, and D, for example, then it's not going to work. It's not going to be accurate. Your reading will be not accurate. So that's why I'm more focusing on biochemical. Okay. So in this case, in this subject, I'll only be able to discuss these three only. Of course, I will introduce to you some other biochemical, but only just some introduction only, not in detail. Okay, so in general, I believe that some of you comes from biochemistry, so you are more well versed than me. So in general, I'll be talking about antibody, DNA, and aptamer, and this will be the last. Okay, I'll start with DNA first. So these are the comparison between these three biochemicals. You can see why we are concerned about the size. Okay. One more thing is why is it we are also concerned about native, and this one is artificial. And you can see the production of antibody. Right now you can produce chemically synthesized, but the common process, the common procedures is using rabbit, still using rabbit. How is you induce the production of the antibody inside the rabbits, and then you harvest the antibody. So it's quite, you know, the process is not that friendly. Okay, but for aptamer and DNA, you can chemically synthesize the DNA. So antibody is big, so it's a combination of proteins, nucleic acids. Well, I hope that you know what is nucleic acid. Nucleic acid is the base structure, the simplest structure, and protein is based on combination of nucleic acid. Okay, so I'm not going to tell you what is nucleic acid. Please find out what is nucleic acid if you don't know. So you can see it's quite difficult to produce antibody, requires animals and harvesting the antibody from the animals. Okay, aptamer, even though it is can be chemically synthesized, but it requires designing. Okay, aptamer requires design. You have to design the aptamer. Okay, I will not be talking about aptamer today, only DNA. So that's why it is mentioned here laborious, because of the designing process. This one is quite easy because DNA sequence, the I'll explain later, that it is already in the system. Okay, I'll talk about it. Okay. All right, so these are among the comparison between these three biomolecules. Okay. But the good thing about antibody, mostly is very specific. It's very specific, mostly. Yeah, not all, of course, but mostly it's very specific. Aptamer can be specific if you design it well. Okay. And DNA is supposed to be specific, but there are some issues with DNA is what we call as non-specific interaction. So just bear with me first. So these are comparisons. As you can see, there are some negative and positive points for all these biomolecules. Okay. So the first biomolecules for today that I would like to explain to you is only on the DNA structure. So don't worry, it's not going to be difficult. You don't have to remember this for your exams, but for you to understand how it works in the senses, okay? Where's the structure? Right? Yep. I don't know why it does not. Okay. All right. So the basic DNA helix structure, okay, I'm sure you know about DNA. So it's your fingerprint, it's your identity. Okay, so this is the bases, okay, the nucleic acid bases, okay, for DNA. So this is where the base pair, the blue one, okay, you have adenine, guanine, thymine, and cytosine. So I'm not a biochemist. I only know some basics. Okay. So these four bases is inside here. Okay. And you have the phosphate out backbone. Okay. Phosphate group. This phosphorus, oxygen. So it's all chemicals. Yeah. And then you have sugar. Sugar, where is it? Yeah, around here. So the backbone, okay? And in the middle are nucleic acid bases, which is in short, ATCG. Yeah, adenine, guanine, thymine, and cytosine. So ATCG. So for us to understand, there is specific interaction between the nucleic acid, between the bases. For example, adenine, it only match with thymine. Okay. In short, it interact with A, interact with T, whereas C with G, as you can see here. Stacking interaction between bases along axis of double helix, size and base content, and sequence. So in the middle here, this is what is happening. And we have hydrogen bond. Okay, hydrogen bond between the bases. I think you have learned before what is hydrogen bond. Okay, so if you understand the nucleic acid structure, nucleic acid usually they have nitrogen and they have carboxylic, C double bond OOH. Okay, so this interact in the between the bases, between the nucleic acid. So this is how you get the chemical structure of the DNA. The most important, you understand that there's only A, T, C, G nucleic acid or bases in DNA. And A will interact with T. This what is formed between what we call as DNA double helix structure. Double helix or double strand structure. Okay. So if you have single strand DNA and single strand, another single strand DNA, they will react via hydrogen bond and they will only interact ATCG. They will know AA in A interact with A or A with C. No, it's AT between CG or GC or TA. Yeah, I hope you grab that idea. And some of you are better than me, of course, in biochemistry. Because why? This is the basis of biosensors, DNA-based biosensors. Okay. So what happened here is, okay, I'm going to start after this. You have, yeah, for example, okay, let me draw a beaker, which is easier for you to understand. And then you have this receptor. Okay, I just want to relate the principle of sensor just now. You have DNA receptor. Okay. Now, this DNA receptor is designed specifically to represent the sequence of tuberculosis. Means that specifically designed to represent bacteria for tuberculosis, Mycobacterium tuberculi, meaning that this is the DNA sequence for Mycobacterium tuberculi. Okay, now I have been coughing for 3 months, for example. And then I submit my sputum, okay, to the one who are going to test my sputum. Okay, after certain process, they obtain a sequence of DNA from my sputum. Okay, and then let's say they put the DNA from my sputum into the beaker. Okay, what happened if we have a perfect match? Okay, we have a perfect match means that the bases, the nucleic acid from DNA from my sputum, and then DNA receptor that is inside the beaker designed to show that it is clear. If it reacts, it means that I have Mycobacterium tuberculi, for example. So if there is a clear reaction, means that all the A, T, C, G, G have a perfect match. Okay, perfect match. Now there is no other conclusion. It is a Mycobacterium tuberculi in my sputum. Okay, so that is how DNA biosensors work. It based on the matching of the interaction between the A, T, C, G. So what happened if you have mismatches? Yeah? Okay, let's say this is the DNA from the sputum, and then when you mix it with the DNA receptor that already in the beaker, and then you have half reaction, and this one 50% no interaction between ATCG. That there is no match. There is no match because the A and the A does not have T to match. Okay, some of the sequence are not matching. So what is the conclusion? The conclusion means that there is a possibility that it is also probably Mycobacterium, but it's not Mycobacterium tuberculi, meaning that it might comes from different same species but different variant probably. Okay, so DNA biosensors it can be very difficult unless you have a very good match. Then you can be very sure that it is positive, for example, Mycobacterium tuberculi. So I think I stop here. I'm not going to discuss a very difficult concept today. But at least you understand up to now is, I hope that you can do some homework to study, to understand the DNA structure. Some of you are from biochemistry, so yeah, it's quite easy for you. And then to understand the nucleic acid, the interaction between the nucleic acid, okay? And what does it mean when I say it match or it does not match? Okay. All right. And then let's say I have given you an example of DNA receptor, because it goes back to what we have discussed just now, receptors. Okay, in this case, we are using DNA. Okay, these DNA receptors has been designed, meaning that if anything that if any other DNA from the samples match perfectly with the DNA receptor, meaning that confirm the DNA from the samples is Mycobacterium tuberculosis, for example, because these receptors has been designed, the sequence has been designed to really represent the Mycobacterium tuberculosis. Okay. Okay, so I hope that you can